Welcome to the new video of our damage mechanism series. In this video, we are going to talk about microbiology influenced corrosion. This kind of corrosion is generally present due to the microbial activity that is happening inside the equipment. Generally, it is associated with stagnant flows. It can also be identified sometimes by smell as well, so it gives you a vibrant appearance. Generally, it is present in tanks, but it can also be subjected to various other equipments also. So, description of damage: the corrosion caused by living organisms such as bacteria, algae, fungi. It is often associated with the presence of tubercles or slimy organic substances. Often the bacteria produces localized corrosion in the form of pitting and crevice corrosion. Affected materials: most common materials of construction include carbon alloy steels, 300 series SS, 400 series SS, aluminium alloys, copper alloys, some nickel alloys. Titanium generally appears to be resistant to this kind of damage. Critical factors: microbiology induced corrosion or MIC is found in the aqueous environments. Occasionally it is found in the surfaces where the water is intermittently present. Stagnant or low flow conditions allow or promote the growth of microorganisms. Because there are many types of organisms that can survive and grow under a variety of conditions, including lack of oxygen, light or dark conditions, high salinity, pH ranging from 0 to 12 and temperature ranging from minus 15 degrees Celsius to 115 degrees Celsius. Systems may become inoculated by the introduction of the organism that multiply and spread unless controlled. Different organisms strive on different nutrients, including inorganic substances and organic substances. In addition, all organisms require a source of carbon, nitrogen, and phosphorus for growth. Corrosion is often blamed on iron oxidizing bacteria or SRB. However, these organisms are typically only the part of complex colonies of multiple types of interdependent organisms, each capable of creating byproducts that might be food source for others. In leakage of the process, contaminants includes hydrocarbon or H2S may lead to massive increase in biofouling or corrosion. Affected equipment: MIC is often found in water-cooled heat exchangers, in bottom water layers of storage tank, in piping with stagnant flow, and piping in contact with some kind of a soil. Product storage tank, water-cooled heat exchangers in any unit where cooling water is not properly treated can be affected. Stagnant process lines with trapped water can suffer MIC. Drain systems and other water containing systems associated with the docks and ship loading unloading facilities can experience MIC. MIC is commonly found in the equipment where hydro test water is not removed or where the equipment has been left outside and unprotected. 300 series SS are often affected unless precautionary measures are taken. Fire water systems can be affected. Appearance and morphology: MIC corrosion is frequently observed as localized pitting under deposits or tubercles that shields the organisms. So tubercles are basically small areas that shield the organisms around. Tubercles also form on carbon steels exposed to the oxygenated water, or so the presence of the tubercles is not the guarantee of MIC. Damage is often categorized by the cup-shaped pits within the pits or halo effect, that is, it is going to be very colorful appearance in carbon steel, or subsurface cavities in stainless steel. However, these pits are often indistinguishable from under-deposit corrosion in the carbon steel and chloride pitting in stainless steel. Pit morphology alone may not be a reliable indicator of the cause of corrosion. Prevention and mitigation: microbes require water to thrive. The system that contain water should be treated with biocides such as chlorine, bromine, ozone, ultraviolet light, or proprietary compounds. Often multiple biocides are needed to keep the levels of bacteria in the appropriate ranges. The proper application of the biocides will control but not eliminate the microbes, therefore continued treatment is often necessary. Periodically flushing and cleaning the susceptible systems will minimize the colony growth and result in MIC. Minimize low flow or stagnant zones in the water containing systems, and flow rate of 3 feet per second or 1 meter per second will inhibit the colony formation and thereby minimizing the likelihood of MIC. Line should be sloped to assist in drainage. The system that are not designed or intended for water containment should be clean and dry. Empty the hydro test water as soon as possible. Coating and cathodically protected underground structures and interiors of the storage tank have been shown to effectively mitigation methods for MIC. Biocides are generally not effective systems that is already contaminated with the colonies protected by a sludge layer. So effective mitigation of the established organization requires complete removal of deposits and organisms, typically using some combination of pigging, blasting, chemical cleaning and biocide treatment. Inspection and monitoring. In cooling water systems, the effectiveness of the treatment is monitored by measuring biocide residual, microbe counts, visual appearance of water. Sampling and analysis can be performed to better understand the bacteria population. While effective treatment and monitoring are key to identify the potential for MIC, it still can occur in the areas with a stagnant flow. Special probes have been designed to monitor for evidence of fouling that may precede or coincide with MIC damage. Corrosion coupons can be used to identify MIC by sampling the coupon surface after it is pulled from the surface. However, it should be noted that just because the sessile bacteria population are not found on the coupon does not mean they don't exist in the system. The increase in the loss of duty of the heat exchanger may be indicative of fouling and potential for MIC damage. RFT can be performed to gauge the depth of corrosion on the heat exchanger tubes. Foul smelling water may indicate biological activity. MIC is often highly localized, so a technique that can find localized corrosion should be chosen as applicable to the circumstances, that is VT, automated UT or RT. Related mechanisms is going to be cooling water corrosion, brine corrosion, oxygenated water corrosion and concentrated cell corrosion. So this pits that you are seeing, these are microbiology induced. Again, you'll also, I have said that you'll see colorful appearance. So these are that appearance that you can see. This is known as a halo effect. This again is a halo effect that you are able to see. So this is fungus that has been developed on the inner surface of the equipment. Again, the same thing, this pit that we are identifying, this is MIC beneath the tubercles. Again, small pits can be seen. Again, these are small pits in the heat exchanger tubings. Same thing. Also, you can see here you can see that these are pits that are developed. This sludge layer, in this you are able to see this colorful appearance. This is algae formation and this is a halo effect that we are able to see. Okay, so these colonies are going to be the microbiology induced damage on the surface. So that's it for this video. In this video we have talked about microbiology induced damage. This is all for this video. In the next video we'll talk about some other damage mechanism for our damage mechanism series. Thank you.